everyone. Welcome to my channel Capsa. Today's topic is gram staining. So, what is gram staining? Actually, it is a staining technique that used to identify bacteria, which helps in the diagnosis of bacterial infection very quickly. So, we can write like staining technique used to identify bacteria quickly. Okay. Now, why the name is like gram staining? Because Danish bacteriologist Hans Christian Gram first introduced this technique in 1882. And he used this technique to identify microorganisms that causes pneumonia. Hans Christian Gram introduced this technique in 1882. That is very much important and used for the identification of pneumonia. We can write like pneumonia identification, okay? Now, in this technique, actually crystal violet or methylene blue is used as primary stain or primary color. And the organism that retain the primary color or primary stain will appear purple brown or violet under the microscope and known as or identified as gram-positive organism. And the organism which do not take the color will appear as red or pink and is known as gram-negative. Okay? So, which retain the color and appear as purple violet or purple brown or violet is identified as gram positive. And which do not retain the primary color appear as red or pink identified as gram negative. Okay. Now, we will understand the procedure step by step. Okay. So, the procedure. In the first procedure, we take an inoculation loop which is used to transfer the suspended culture to the microscopic slide we are using. Okay. So, we can write like transfer the culture into the microscopic slide or slide by using inoculation loop. In the second point, culture is spread with the inoculation loop to form an even thin film or known as smear. So, spread the culture. And in the third point or in the third step, fix the smear and dry it with the help of gentle heat. Okay. So, we can write like fix the smear by using gentle heat. Understand? And why we use this heat? Because heat helps the cell to adhere to the glass slide. Okay. And in the fourth step, we use primary stain or crystal violet color over the smear and wait for few seconds. It can be 10 to 60 seconds. And excess stain is rinsed with water. Mind it, the goal is to wash off the stain, not the lossing of culture. Okay. So the primary stain is applied. And rinses after few seconds. Okay. And in the fifth step, we use iodine solution to fix the dye or primary stain or the crystal violet color. Okay. And after that, after few seconds, we rinse with water. So iodine is applied to fix the primary color. And it forms a complex that is known as CVI complex or crystal violet iodine complex. Okay. And in the sixth step, we will use few drops of decolorizer. It can be ethanol or acetone over the smear. Okay. And after few seconds, rinses it. So decolorizer is added. It can be ethanol or it can be acetone. Now, why decolorizer is used? Actually, it's used to remove the primary stain or crystal violet color. Now, we need to remember some tricky note that in gram-positive bacteria, peptidoglycan content is high. But in gram-negative bacteria, lipid content is high. So, when we add decolorizer, CVI complex that is formed do not remove from the peptidoglycan cross-linking, but due to higher lipid content, CVI complex dissolved through the lipid layer and removes in case of gram-negative. Okay. So, if this is the peptidoglycan layer of gram-positive bacteria and this is the lipid layer of gram-negative bacteria, then CVI complex will retain in the cross-linking of the peptidoglycan layer. Okay. But CVI complex in case of gram-negative dissolves and removed from the layer. Okay. Now, in the seventh step, what we use? Actually, the smear is counterstained with fusin or safranin. And after 40 to 60 seconds or few seconds, washed off with water, then air dried the slide. So, counterstain is applied and then air dried the slide. And last, go for the microscopic examination. Okay. And if you see under microscopic examination, the color is violet or purple brown, that is gram-positive bacteria. And if the color is red or pink, then that is gram-negative bacteria. So this is the end of my lecture. If you like my video, please do like, comment, share and subscribe.